Welcome to the Genomics of Energy and the Environment, JGI user meeting. We're in a period of very dramatic changes in genomics. The landscape is changing. It's driven by, enormous technological changes. Rarely do you have a field of science where the metric has increased a millionfold in less than a decade. And I think that's happened in genomes or genomics. And in response to that, the JGI, with support of DOE, has been developing a strategic vision for what we become in the next decade. And one of the good things is that DOE is very determined to have a genomic user facility. And so it's really a vision of how we evolve, but it's not as though we go away, which is good. And so where we see, and on our website, there is our strategic vision, but we will continue being a next generation sequencing facility in that there are lots of problems that need genomes for. And there are certain kinds of ones that the JGI can uniquely accomplish because of our technologies as well as our abilities and analysis of data. But I think a major area where the JGI will focus in the future is the conversion of sequence to function, to be able to sort of cross that or try to diminish that giant gap between our ability to generate sequence and our ability to derive important functional information from that sequence. Sequencing is a monolith. It's just one structure. There is not a single functionator. And so there's not a way to push DNA at one end and get function at the other end. And so we will have a variety of capabilities that are suited for a large-scale facility of the JGI, which I imagine will evolve over time. I think one of the capabilities that we are developing and have user programs in is the ability to write DNA synthesis, connected to synthetic biology and pathway engineering. And I'm very pleased that today's keynote speaker is Chris Voigt, who has really been a pioneer and luminary in this area. Chris received his PhD at Caltech with Francis Arnold. He then went to work with Adam Arkin at UCB and LBNL for a short period of time. Then he took a faculty position at UCSF. And about a little over a year or 2 years ago, he was seduced away to join the biological engineering department at MIT. The field of synthetic biology, for society to say that this is something crucial, it needs some examples of how it has impacted biology. And I think it's one example is artemisinin. It's sort of, in some ways, the singular example, but it's an important one. And artemisinin is now going to be sold next month, or sold very cheaply because of synthetic biology. Chris, I think, is working on a project with regard to nitrogen fixation that has that kind of appeal, that you want this technology because it can contribute to society and provide something crucial, the ability to fix nitrogen for agriculture. And so Chris is going to be talking about mining sequence databases, accessing multi-part cellular functions. It's a pleasure to have you here.
Great, thank you, Eddie. It's absolutely fantastic to be here. I'm really excited about it. And part of that is because JGI is so well positioned right now to have a tremendous impact in the field of synthetic biology. What you see across the world, really, is that all of the top sequencing institutions are retooling themselves for synthesis and for synthetic biology. You have JGI, which is making serious efforts there. I'm running a facility that's going to be part of the Broad Institute that is looking at some of their systems. You have JCVI, of course, has been doing this for some time, as well as international ones like BGI, I think just put $200 million or so into synthetic biology. And so it's a fantastic time to be here, and I'm coming with two hats. One hat is somebody that's running a synthetic biology facility in another sequencing center, and the other hat is as a future user. What types of things that a user might be requiring out of sequencing/synthesis facility. And what I really want to drive home with that, and I'll come back to it a number of times in the talk, is that sequencing is not enough, synthesis is not enough, that you really need design on top of that in order to create useful functions. And so, of course, I don't have to tell you how fantastically successful sequencing has been over the last couple decades. And as a result of that, what we've done is populated the sequence databases with hundreds of millions, if not more, genes. And so there's this fantastic amount of sequence information that's been deposited, starting with the genomes of microbes and then going to these deep metagenomic sequencing that's just populating all of this genetic information that's there. But now if we really want to utilize the sequence information that's in these databases, we have to close the loop. And this is where synthesis and then ultimately the screening of function is what allows us to really understand what's there and ultimately to harness it. And so we've been doing this for a number of years in my lab, where we started out by looking at enzymes. So for example, this is a project that we did a number of years ago where what would have been done in biotechnology previously is if you had a single enzyme, you would take that enzyme and just keep hitting it with a hammer, even though there's all this information in the database around alternative enzymes that you could be using. But what we did is we worked with DNA 2.0, which sells DNA synthesis, and we printed out every single example of that enzyme that existed in the database. So this was a library of about 90 enzymes from all walks of life, and then you could screen all 90 enzymes for the particular function that you wanted. In this particular case, we found that the function that we wanted was either out of swampwort, which is a plant that lives outside San Francisco, or bok choy. And for the particular metabolic pathway we were looking for, that was totally non-obvious. And so now we've done this again and again because it's just so easy. You know, in the old days, you would run a blast search and you'd kind of use annotation and homology to try to figure out what's going on. But now you can take that blast search and just cut it out, print it in an email, send it off to a synthesis company, and just send you every single sequence within that, as long as you've got the cash to pay for it. And so this was '89. There have been now a number of different labs that have been doing this. I have a paper with Jay Kiesling. Last year we were looking at cellulases and pulling cellulases from a wide variety of sources and so on. Also things that are more complex, like regulatory proteins. One of the things synthetic biology is very interested in is building genetic circuits. And at the core of that, you have to have transcription factors. And we have been building libraries of hundreds of transcription factors, trying to identify ones that can be used together. Sometimes it's even just very simple parts. I just finished a project that I hate at the irritation of my postdoc, where we needed a bunch of terminators, transcriptional terminators, and that'll be clearer as I go on as to why. So we had to print 700 terminators and screen them for function. I mean, just imagine how many terminators are in the libraries of metagenomic sequences that JGI has put together, or ribosome binding sites, or promoters, and so on, and how much utility could be derived from those. So now, the talk that I'm going to give is actually stepping up beyond this, where what's common about all of these different projects, whether it be simple parts like terminators, or enzymes, or transcription factors, is that they're very simple parts or single genes. And the thing is, if you want to screen a library of 90 enzymes, then you need only the simplest synthetic biology to guide you, in the sense that all you need is an expression vector. You drop the 90 genes into that expression vector, and then you screen them for activity. And so there was not a lot of choice or decision or design that had to go into that process, but still it's a start. But what if you really want to scale up? And if you want to say enzymes are interesting, and we can learn a lot from that, but what we want to do is an order of magnitude larger and go after functions that are actually encoded by many genes and perhaps hundreds of parts. And so this is where we started looking at gene clusters. And gene clusters are simply units of DNA that are in the genome that have all of the necessary genetic elements that are required for that function. So for example, I'm showing for nitrogen fixation, which I'll show a lot in this talk. That's a 25,000 base pair unit of DNA that has about 20 genes that are required, that are necessary and sufficient for nitrogen fixation. And there are lots and lots of gene clusters out there that encode all sorts of interesting functions in bacteria. So another system that we work with is protein secretion, which requires about 34 genes and 35,000 base pairs of DNA. There are lots of those that involve cellulosomes. There are photosynthetic gene clusters. And then there's this enormous capacity for small molecule synthesis. There are a lot of gene clusters that involve all of the genes necessary to build complex natural products. And actually, in just that alone, there's an extraordinary capability. So I worked very closely with Michael Fischbach at UCSF, and he recently ran some analysis of sequenced genomes, and he was able to identify 20,000 gene clusters in those sequenced genomes, of which only a tiny fraction are known as to what molecules they produce. So within those gene clusters, there are antibiotics, there are insecticides, there are herbicides, there are anti-cancer, anti-tumor. There's all sorts of molecules that are present there. But how in the world are we going to go into those sequence databases and be able to actually access those functions? In particular, in the metagenomic samples, where you may not even know what organism that sequence came from. You know, how do you wake that back up in a foreign organism so that you can access the functions and the diversity that are there? So what you need, and I'll keep coming back to this, is three things. So if you want to be able to access the function, of course you need sequencing, you need bioinformatics, like what Mike has done, just so you can identify the sequence and you can get some prediction as to what the function is going to be. But that's not enough. You also need synthesis because the DNA sample is long gone. It's just information in a database. So you have to be able to access that information back out again. And to do that, you need to be able to synthesize the DNA. But the synthesis is not enough. And the reason the synthesis is not enough is because you're not putting it back into the organism from which it came. You've got to be able to make it work in some new organism, some host. And so for that, you need synthetic biology and design. And I'm going to give you a sense of how deep that is. And when you're approaching a problem like this, the real challenge is that genetics kind of suck in the natural organisms. The way that these functions are encoded by their hosts is incredibly complex, I would argue largely unnecessary, and really confusing. And so you have to get rid of all of that. And I love to use this one example of one of the systems that we study, which is type 3 secretion in Salmonella. And type 3 secretion in Salmonella is something where it's very important for that organism to be able to turn on that function at precisely the right point of an infection and then turn it off at precisely the point at which it needs to turn off. And so it's got this incredible regulatory network so that it does that properly. Now, we have a collaboration with a small company called ProNutria that wants to use these devices to export nutraceutical proteins. But for that function, you need to break all this regulation. But it's so redundant, it's so embedded, and it's so thorough throughout the genes that just disrupting one interaction is not enough. And often you don't even understand how it's coming back and stopping the function that you want. So that's problem number one. The regulation's complex, it's not understood, and it's difficult to get rid of. The second problem is the actual literal encoding is something that we would never have come up with. So this is an example of one aspect of this gene cluster, one of the operons. And this is an RNA-seq experiment. And all I'm showing you here is where the promoters are for this region of genes. And it should strike you right away that the promoters are all over the place. You open up Nature Reviews Microbiology and you look at the gene cluster, it looks very nice. There's a nice promoter up front, it turns on this operon, there's a promoter down there that turns on that operon. but it's a total mess. You've got promoters pointing in the wrong direction in the middle of genes. You have multiple promoters upstream. You have promoters going against the gene order for no apparent reason, and so on. And every time when you look at this, if you want to make a substitution, if I just want to change a gene, or change a ribosome binding site, or change a promoter, there's all of this embedded genetic encoding that you're changing on top of the mutation that you actually want to make. And that makes it really hard to make modifications to do what you're expecting to be done. And so we looked at this problem, and we said, well, we still want to have some platform where we can approach these complex multi-gene functions and so on. Maybe we can turn a little bit to some of the basic concepts in synthetic biology to kind of help us understand how to manipulate a pathway like this. So to do this, I'll just go over very briefly the basic hierarchical organization of how synthetic biologists think about genetic systems. And so at its very core level, we have things referred to as genetic parts. And a genetic part is a unit of DNA that has a biochemical function. So a promoter might be a genetic part, a ribosome binding site terminator, and so on are genetic parts. A gene might be a genetic part. And then we take those basic units of DNA and we combine them in order to create what's known as a device. And a device is just a set of genetic parts that produce a user-defined, human identifiable function. So you might have a genetic circuit that performs an electronic function, a metabolic pathway, something that we can convey as humans. Then you take the devices and you connect them up in order to create a system, which is just a combination of devices that performs a task, and then you put that in a chassis. Okay, so that's the lingo. And what we're going to do to apply this lingo to approaching gene clusters is we're going to turn it on its head. And so this is a process that we refer to as refactoring. And that's a term that is taken out of the software industry, where if you're having a problem with a piece of code, you might refactor it. So if Microsoft Word, it keeps crashing on you, one approach is to say, okay, we've stopped fiddling with the code to try to remove the bugs. We're just going to rewrite it from the ground up to be more modular, to be more engineerable, to be more robust. But to an observer, it's going to have the same function. So the underlying code's rewritten. To the observer, it has the same function. And so what we're going to do with respect to gene clusters is the same thing. We're going to rewrite the DNA code to be more engineerable, to be better understood, to be more robust. But the overall function is going to be identical. And so the way this works is the following, where we start out with the gene cluster on the computer. And so we have the sequence information. So this is just pulled out of a database. We're not necessarily working with the organism. We then eliminate all of the non-essential genes and the non-coding DNA. So we just get rid of it, and we never come back to it. Then we take each of the open reading frames. We go through a process called codon randomization. And that's different than codon optimization in the sense that we're not trying to kind of optimize expression. What we're trying to do is to identify a DNA sequence that is as far away from the wild type sequence as possible. Okay? And the idea there is we want to blast out all those promoters, internal RBSs, all of the regulatory elements that can be internal to a gene. So then we're left with a bunch of open reading frames with completely different DNA sequences that we then believe are inert to all regulation and don't do anything except encode that amino acid sequence. So then we organize those open reading frames into artificial operons that make sense to us as designers versus the way that they're naturally organized. Create synthetic parts where you control the levels of expression by selecting ribosome binding sites and promoters and so on, all synthetic, all fully characterized, often with mathematical models behind them. And then the last step is you create a so-called controller, which then replaces all of its regulation that was there in the host with whatever synthetic regulation, however you want it to respond to the environment, turn on, and so on, integrate signals. And a lot of that is the classic synthetic biology. So this is a completely radical reorganization of the natural genetics, with the idea of creating a system that's effectively genetically solved, in that you understand every part is there and what its function is, and it has such a different DNA sequence that you've got to use a whole gene synthesis in order to access it. Okay. So that's the introduction of the talk. So we're going after gene clusters. We're refactoring them with the idea of being able to transfer them into new organisms. And this is the sort of pictorial outline of what I'm going to go over for a specific system next. And so the whole idea is that we want to take some function that's encoded by a number of genes from one organism and move it into a second organism. We know because of the crazy regulation, the crazy genetic encoding, that often you can't just literally take that DNA sequence and move it in. So instead, you've got to go through a systematic series of steps to start from the sequence information in one organism and end with something that works in the second organism. So the first step is this process of refactoring. So within the starting organism, if we have that luxury, we go through this process of replacement where we substitute all of the native parts for those that are synthetic. We completely reorganize, resynthesize the system. And as a result of that, we very quickly lose activity. So we start out with a nice wild-type cluster. The engineerable version, it makes sense to us, but it's also lower in activity. And it's so low in activity that you now can't transfer it. So then we go through a process where we optimize that function within that organism to build it back up. Then we transfer it into the new organism, and it loses activity again. And then you have to go through a final step where you re-optimize the system for that organism. Okay, so it's refactor, get parts that are going to work in the new organism, put them in, get them substituted. It causes the system to fail. You then have to optimize. Then you can transfer. Then you've got to re-optimize. Okay, so those are the steps. Now, the system that I'm going to talk about is one that we've been fascinated for a number of years, and this is nitrogen fixation. So this is where a number of organisms are able to take atmospheric nitrogen and convert it to ammonia. And this has been a long-standing sort of challenge for biotechnology, where the dream is to be able to take that function of being able to fix nitrogen out of one of the microbes that is able to do it naturally, and either put it directly into a plant so it can create its own fertilizer, or put it into an organism that can associate with a plant. And so the organism that we're going to work with is a model system for nitrogen fixation. This is a bacterium referred to as Klebsiella oxytosa. And I'm showing the gene cluster by which the nitrogenase is made. And I just want to make the point that it's actually, so it's an individual enzyme that performs the nitrogenase function, but there's a very complex and very delicate biosynthetic pathway that creates the FeMoco cofactor that gets loaded into the nitrogenase, which then performs the function of interest. So it's a very challenging gene cluster. And one of the reasons it's been recalcitrant to being transferred into a new host is that it's very finely balanced, and you'd have to move 16 genes in order to get it to function. And getting that into a chloroplast or into a rhizobial organism is very non-trivial. So now our first challenge was to go into the system, refactor, reorganize it, so that we're replacing all of the components with those that are transferable. And so a year ago, we published some work where we published the initial refactoring of this gene cluster. And this is this radical reorganization. So now for real gene clusters such as this, we go through this process. We eliminate non-coding DNA, recode all of the open reading frames to be as far away as possible from wild type, remove all the transcription factors, so all of the things that are regulating it with respect to oxygen and ammonia. We're just left with the open reading frames. We organize them into operons that made sense to us, replace them with synthetic ribosome binding sites and terminators and phage promoters, then built a controller that then tells that cluster when to turn on, synthesize the whole thing, And after 6 years to make this slide, it indeed worked, but pretty crappily. So at the end of the day, after we had gone through all of this process, it's actually incredible that it worked at all, but the version that came up with at the end, the first version, was 7.3% of wild type activity. So wild type's 100% activity, and we're able to recover 7.3, so about a 20-fold-ish reduction. But we were so tired of fiddling with the system, that's what we published at the time. And let me give you just a very brief feeling for why it took so many years to actually get through this. So we started this project in 2004, and there were really no tools for us in 2004. We had to make up everything in the lab as we went. And so what we would do is we were very ambitious. That's when I started my lab at UCSF. And we would go in, and I knew the DNA 2.0 guys from when I was an undergrad. I was in the same lab with them. And so we would talk, and I'd get really sort of ambitious, I guess is the word that I kept being told. And we would we would take a single shot at it. So we'd go through this process. We would re-engineer each of the open reading frames. We'd select ribosome binding sites from God knows where, put it together, one long operon, and then just send that off for synthesis, get it back, and lo and behold, it would just fail, just zero activity. This one's actually not bad. For the type 3 secretion one, I built a 14-gene pathway all at once, absolutely no function. It was like $35,000. It was just depressing. It was bad. So then you have to figure out, okay, what's wrong? What's in that giant cluster that didn't work? And you can imagine that you make all of these changes, and as a geneticist trying to figure out, going backwards, what of those numerous changes caused the whole system to break down. So you would do some debugging. So this is just one example. I'm trying to give a sense of scale. So we would build 5 at a time, roughly. And so in this case, it failed because the gene we had designed was filled with transposon insertion sites. So that takes a while to figure out. You put that on the blacklist of don't go to that site anymore, and then you resynthesize the gene. Then you put the new gene in, and that seems to work. And then you've got to do some tuning. It's way below activity, so we would build a library of about maybe 5 to 10 variants where we would change the RBS sequence, change the promoters used, whatever we felt was necessary at the time. You'd go back, you get this small increase, and then you say, okay, well, that's done. And we ended up having to do this over and over again. Just you would get NIF-H to work, then you'd get NIF-D to work, but then NIF-H would fail, then you get NIF-K to work, you realize you really needed NIF-Y. I mean, it was just this cyclical issue as we were moving through this pathway. But the emphasis here is that we're doing about 5 at a time. Single shot, debugging 5 at a time, correction 5 at a time, and then you got to loop on that design, build, test cycle over and over again to figure out where you are in the system. And then finally, you would give up, and this is the point where we gave up. And so here I'm showing where we were turning the cycle on independent pieces of that cluster, and the percentages are where we got with that piece until we gave up. So at the top there, the HDKY operon, we gave up at 30%. BQ was particularly tough. We gave up at 10%. And so on, but then we'd have these refactored units. And then we turned to a method that was developed by the Venter Institute in order to assemble larger pieces of DNA, which around 2007, 2008 became available. And then we could assemble these relatively rapidly. It would take about 6 weeks into the final cluster. So this is actually the version 1.0, which did not get published. This is where Karsten ran into my office, enthusiastic that he got some function, but I was sitting there very awkwardly at my desk holding the paper sideways, trying to see whether there was a bump in the curve or not. Sort of told him we got to do a little better than that. And so then we'd have to go back to the beginning and restart turning the design, build, test on the individual units. And actually, when we did that and we went back, the interesting thing here from your perspective was that we were doing everything in Klebsiella, but like I was alluding to at the beginning of the talk, we were just basing everything on the gene sequence that was in the database. So we just went, and anytime we had something synthesized, we'd just get it synthesized from what was in the database. So it turns out in this 30 kb region, there were about, I think, 27 errors in the sequence, of which 13 hurt function significantly and 2 destroyed it. So in this case, we were using the information, but it was not of a quality that was allowing us to move forward. So we had to re-sequence it and get the depth that we needed and so on. I think it was also sequenced in like 1983 or something, so yeah, times have changed. So then we found those problems, we fixed them, we go back and resynthesize, and that's where we got 7.3% of activity. So after going through all that effort, we said, great, proof of principle, let's get it out there. So now, but we were reflecting about this as we went forward. We were saying, well, this is getting very, very frustrating. We're not actually achieving what we set out to do. And so we've got to rethink the process and perhaps pull in a couple partners to help us with it. And so we decided we kind of could take 2 approaches. So there was this thing where every time we thought of something to try or we found a improvement to one of the earlier stages of the hierarchy, you got this individual piece to work better, and we had to wait around 6 weeks to build this 30 kb cluster, and then you had to characterize it and so on. So the design build test cycle was about 5 at a time. You'd build them all, you would test them, and then you'd loop back, and that would take this long period. So we could go about accelerating this process by being faster in design, faster in building, faster in testing, and just go through more cycles iteratively. What we decided to do was a slightly different approach, was to go after the system in parallel. So the idea is that instead of designing one, we wanted to start designing hundreds and ultimately thousands and tens and thousands as we're moving forward. Then take those permutations, screen them all for function, take that information and learn from it to guide the next round of design. So instead of going faster and faster around the cycle, you're spreading out, you're taking a lot of permutations and a lot of information, and then you're using that information back. Okay, so that sounds great, but actually every single step in our pipeline, we had to go back and improve from the design to the analysis in order to actually be able to do this. And one of the first things we realized was going to be a big problem was just the very simple aspects of design. So I've got a little mark there that says also the trivial, because it's great to talk about all the computer-aided design innovations and learning algorithms and so on, but sometimes just building the primers associated with a 30 kb, 100-part system that you're trying to assemble, much less 10,000 of them. Or taking all of the sequencing information in and verifying that you have the right construct. There's some things that I'm sure we don't take for granted, but where if we're really going to scale it up, we need to take a fundamentally different approach to design. And luckily, at about the same time that this was an issue, I was starting to develop more deep collaborations with a fellow at Boston University in their electrical engineering department. His name's Doug Densmore. And what Doug had come up with was a language that looked absolutely insane, like nobody would ever adopt it as a mode of genetic design, but it had one feature about it that was fascinating to me. So what Doug did is he said that this whole concept of the way that we design a genetic system by going into Vector NTI or Ingenious or APE or whatever it is you do, and sitting there and putting in these units of DNA according to non-existent design rules was actually really shooting ourselves in the foot. And what he did instead is he created a language, and some of the basic functions are there, where instead of sitting there and putting in DNA, what you would do is you would define the parts, thinking about the lowest level of the hierarchy, and then you would constrain those parts until you came up with the design that you wanted. So you would have to articulate in a language that he developed that the promoter has to be in front of the RBS has to be in front of a gene, there has to be a terminator after. You would have to articulate all that as the designer. And one of the most sort of interesting things he did as demonstration is he came over to the lab And you know, I have a fantastic cloning postdoc type who went through and he listed on the board every single thing that you can imagine as a genetic designer that you would want to be encapsulated as a rule in a language like this, and he could convert each one of those into one of these rules, right? So anything that you can imagine as part of design, as an experimentalist, you can capture using this language. And so it's just another way of specifying a design. And this is an example out of synthetic biology. So on the left, I've got the repressilator. That's how we all think about it. That's just a vector with all of the parts in that vector. The exact same specification for that design is on the right. And it looks like a mess, right? So it looks like a programming language. But it's actually way more trivial to go in there and say, pull out this part and substitute that one. It's a line of code. It's not this kind of straight, you know, you put in the piece of DNA and so on. And then you've got it constrained by rules. And what that allows you to do then is actually relax those rules. So the most fascinating thing for me as I was playing with this is that when you take a gene and you just want to express that gene and you drop it in an expression vector, you're making all these design choices that you don't even appreciate. You know, the whole thing could be popped out and flipped around and put back in. The antibiotic resistance could be over here. The origin could be over there. There's no reason why this particular configuration. And you're just making all these design choices because of history. Here you can formalize that and only make those choices you want. And it's actually really hard to constrain it to a single system. And then what you can do is by lifting out a particular rule, you can then permute to create variations that capture everything that you understand about the system, okay? And so I use an analogy with trying to find a Shakespeare sentence with that, okay? And this will make sense on the next slide. So you can think of, if you're trying to create a sentence and you're trying to find Shakespeare, that one way you could do it is you could take all combinations of words, okay, and you end up with kind of nonsense. Iron close, pat, adhering, behind, study. Doesn't make any sense. You can then constrain the space by applying grammar rules. And that's assigning things like verb and noun to individual words, so at least you make a sentence that makes sense in English. So, you know, the wife angers the monkey. Then you can add constraints, you know, to always before be and so on. You can learn additional constraints if you search and apply those constraints. And all the time you're decreasing your search space for that sentence of Shakespeare. Okay. So now what this language allows that Doug came up for is here's an example of a gene cluster fully defined according to this language. If you were actually looking at the code, underneath this part, you would see all the DNA sequences for all the parts. And then what you could do is write a little script, like at the bottom there, that then permutes according to those rules. So in this case, we just have the words. So we just say, okay, we've got these parts. They define a cluster. Just give me all variations of those parts. And just like all variations of words give you nonsense, all variations of parts also give you nonsense. So you get 2 genes in a row with no control. You get 3 RBSs in a row. You know, 4 terminators, promoter going nowhere, and so on. And then you can start to add rules into the system. You can say, well, I don't want that nonsense. So at least make sure that every gene in my system can be expressed in theory. There's a promoter, there's an RBS, there's a terminator, or it exists in an operon. You can write that formally and then re-permute. And now you get clusters that are kind of already starting to look like they make more sense. And then you can apply any arbitrary rule you want on top of that. So just as an example, the operon rules. So you might say, well, I know for sure that these 3 genes must be part of an operon. That gene must not. But you have to be careful with that, because 99% of the chance you have no idea, right? And you're just, so we've found certainly that floating things we truly don't know is usually pays off. And so in that case, you get all of, you get sequences that are a bit more constrained, but still you can see there are a lot of ways to put that together. You can keep constraining it more and more and more and reducing the search space, but it's really hard to come up with one. Now the great thing about Doug's work is that it doesn't just make a picture for you or come up with a construct. It then feeds into the assembly pipeline that we've set up, where it then creates the control code for the robots that are necessary. It creates the protocols for the steps that are necessary. It goes through everything that's involved with constructing all of those variants, not just designing them. So now I'll simplify this just to describe a little bit of what we're doing in practice to build these clusters. We have a very hierarchical approach where we start off with one method that's very good at combining little pieces, so like a small promoter and a small RBS. We then have PCR-type methods that build out the transcription units, and then we've got Golden Gate assembly that builds out the design. And actually, it's a huge computational challenge to say, because we're specifying everything on the bottom level, right? So we say we want these 100 clusters. And then it's got to figure out, well, what are the minimal number of starting steps? How do you put those together in the optimal way at the next layer? How do you put those together in the optimal way in order to get that final set of constructs that are being built? But once you've got that infrastructure, you can really start to play with it. So I'll just give you some very simple examples of some highly constrained systems and then start to relax those constraints. So going back to the nitrogen fixation system, we started out and we felt this is a really sensitive pathway. We've had a heck of a time over a long period of time, and we know you can't fiddle with it. So we're going to be very constrained. And so the first libraries that we built did things like change the promoters, kept the overall, very constrained compared to wild type, kept the overall organization right, maybe some terminator and some spacer changes and so on, but kept the architecture the same. So this is an example of one of those libraries that came out the back end. This was about, we always make 100 at a time, so whenever you see a number less than that, you know that's the number that failed in the assembly. And so in this case, we weren't that good yet, and we still were able to find a couple that were improved, but modestly so. So then we went in, and we decided to make another library that was a little bit more complex. So we started adding promoters where there shouldn't be promoters, having a lot of variations in strength, various ribosome binding sites, terminators, and so on. And then you go through the process again, assemble them. And here actually something should be striking in that there's obviously something that was done wrong at the bottom. So there's something that killed activity, and you can go in and extract a rule for that. And in these very simple libraries that we were building, we can do that by eye, just as good designers. You can go in and say, well, this ribosome binding site's something that it's very sensitive to, so we shouldn't fiddle with it, and so on. In the more sophisticated things, we're having to use machine learning to help guide our eye with that. So then with that first part of the cluster, we were able to learn from those first two libraries, and we figured out that there was one key ribosome binding site that really mattered. We have a computational method that allows you to scan that strength. We found very quickly one that fully recovered activity. So for that cluster, we got 100% activity after these methods. Okay, so that worked well. We went back, we fixed that one cluster, and so we thought, well, let's do another one that was problematic. So the HDKY operon worked at this point, and so we went back to the USVWZM. So it's a 6-gene pathway, a little bit bigger. It's very sensitive in our hands, and it's involved in FeMoco biosynthesis, which is the cofactor that's critical for nitrogenase. So now we got a little bit wackier with it, and it'll take 2 slides to show you how kind of crazy the genetics get. But what we did is we floated a lot of things. We said we're not going to constrain it at all unless we know for sure that there's a rule under there that has to be preserved. But anything else, operons are important, we'll just lose the operons. 5 promoters in a row, I don't know why that really matters, right, that not be there. So why constrain against that? And really float and be very specific to only incorporate those rules that we truly felt were important. And then this is an example of what the assembly looks like for a pathway of this complexity. You have a bunch of very simple parts like ribosome binding sites and promoters that get assembled first. Then it's a separate method of how those transcription factors get put together in order to create expression units, which then ultimately get assembled into all of the different clusters. And again, you're starting on the bottom level with design. Everything above it is being figured out for you. So this shows the result of that assembly and some of the screening that was involved. So we called this the Stata Library because that's a building at MIT that looks like a mess. I teach in it, and you get nauseous because it's all weird. And so we screened this library, and we were amazed, given all of our issues that we were having with this system before, as to how many solutions there were to the problem and how different those solutions looked from the wild type or the first refactored version. So we changed things like gene order, gene orientation, so we were flipping which direction the genes were going, operon, occupancy, which genes were in which operons, and so on. Promoter strength, how many promoters? We would have a bunch of promoters upstream for no apparent reason. How many terminators? He'd have 3 terminators there, again, no reason. And RBSs. But what's really important here in this graph is that at the point that we ran the assembly and did the experiment, every single one of these constructs, for all their complexity, fully captures our information about how the system should be designed before we did the experiment. So they're all equally designed, even though they're so incredibly different from each other. And that's really a key point. So just to give a quick comparison for this, so with the wild type cluster, of course, we have 100% activity there. And there were some complexities when you look closely within that wild type operon. So there are a couple internal promoters that are typically not advertised in the Nature Reviews microbiology pictures, and there are overlapping genes and some internal feedback loops and so on. And so then when we refactored it the first time, we were pretty conservative with respect to the genetic organization. We kept them all in an operon and so on, but we lost 70% activity. And all the biologists said, well, that's because you threw out all those genetic elements that must have been important. That's why you lost the 70% activity. But then when we went back and just created 100 more designs, of which these are the 5 best, that all have exactly the same amount of information in them as the one that we refactored the first time, we found lots of variants that were doing just as well as wild type. So the second one on that list at 105%, is still in an operon, so it's just selecting better parts. So if maybe we had kept the overall organization the first time the same, but had tried more things, it would have worked. But the one above it is crazy, the one that worked the best. You've got promoters and genes running every which way. You have multiple promoters internally, and so on. And those two, again, have the same amount of design information in them at this point. Now, once you've run the library, you've learned so much that you can go back to that. So the other thing that we tested, because I thought the clear answer to this is going to be that maybe that top one has higher activity, but I bet if you put all those genes in the same operon together, it'll be more robust. So maybe over a larger range of expression levels, you'll be able to preserve that activity. And so we went in and tested the robustness. The wild type system is really not very robust when you look at it. Things have to be very finely tuned. The first refactor system was worse. But then if you look at the top one, that actually preserved activity over essentially all the expression levels of polymerase that we put. So it's actually a much more robust system. And the next one down, which preserves the operon order, reduces the robustness. And so we went back to all of the different clusters we have designed, and we just got this information. And we get just about every robustness profile that you can imagine. You get somewhere at low levels of polymerase expression is optimum, high levels are optimum, you get robust, you get fragile, you get every variation. So there's just a huge amount of information in there that we want to extract so that we can go back and in the next design start to fill out the rules as far as what's really important in this cluster. So just then taking all of these different pieces together, this is where we are as of today. So when we go back and we start to put the different pieces, we're able to recover activity with the HDKY, able to recover activity with USV WZM, as well as BQF, which I didn't talk about. And we just have 2 more genes to optimize, and then we will have recovered activity, but in this completely different, genetically wildly reorganized system as compared to the wild type. So now, at the same time that was all happening, I sort of set it up that we were doing this whole gene assembly, the whole cluster assembly, where we're going after 16 genes simultaneously. So we sort of divided up at the beginning, and we started to do the permutation experiments with the cluster, just going back and trying to fix some pieces that were there. And then we decided to do the whole gene optimization in a foreign host. So for this, we started with the version 1 of the refactored, and we moved it into E. coli. And when you move it into E. coli, you get a dramatic reduction in activity. So it's functional in that host, but you lose a lot of the activity. And what we wanted to do is then go back and doing whole cluster assembly, try to optimize the system post-transfer. And so I don't have time to go into the details of each of these methods, but what I want to point out is that once you have a system set up where you can very rapidly design a lot of constructs, assemble a lot of constructs, and test them, what algorithm you apply on the front end to help you with the design process is almost irrelevant. You can do anything you want. And so in this case, we did design of experiments to select ribosome binding sites. Again, I won't get into the details here. Ooh, it's frozen.
How about, people want parts at very precise activities, and you're sort of taking scrambled eggs kind of approach to it. And so you just need ones with various activities. You don't, because you're making no assumptions of what's going to work.
No, but you do need to have some characterization of what they are.
Like higher, lower, but not exactly.
Higher, lower. And that's part of solving the genetics for one of these systems, in that at the end of the day, the map that you have is not just reorganized, but every part has a defined function. So you can select this ribosome binding site, and you know it has an activity of 1,250.
Right.
And this promoter, it has its activity, and the terminator, and so on. So part of completely specifying the function of a system involves the actual understanding of what the function of each one of those parts are.
Right. Other questions?
Any other questions? Oh, that's not bad.
Jim, this one back here.
You got it?
I see. That's fine. You're actually on the right slide.
So you're stumbling to design principles more or less the same way evolution, or the same way evolution is doing it, by trying a bunch of stuff and figuring out what works. Is there a role for comparative genomics in organization of some of these complex parts, and can you glean something from the multiple promoters that might be in the wrong orientation, not just in this organism but in other organisms, that sort of thing?
Yeah, so the closest analogy to studying natural genetics of microorganisms would be neutral evolution, which has been characterized a lot for individual proteins. But of course, the gene clusters are undergoing these changes that would be considered neutral. The challenge in actually learning from that is that so little is known about the natural gene clusters that you can't look back and say, well, that had 2 promoters in it, so that's why I'm getting 2. I'm not even sure it would work out that way, but the information's not there at all. And even the most highly studied ones have such little information. I think that when you take an alignment of a lot of clusters that do the same things, you see a lot of the variability that we see with respect to orientation and order. So it may be that they're all members of operons, but there are only 2 genes that always have to be together, and everything else is just all over the place. Right? So there might be a connection with that. I certainly think that we worship the wild type sequence too much, and that in the sense that there's this feeling that every base pair must be there for a reason, and if you've changed it and it still works, it's just because you didn't run the experiment under the right conditions. I'd say the vast majority can float, and there's a core set of underlying rules that have to be there, and I think this type of approach, or doing bioinformatic analysis of lots of related clusters out of different genomes, are ways to get at what the underlying rules might have to be. So with that, thank you. So back to getting it to work in E. coli, and So in this case, we're using design of experiment assays, which aren't really important in the details, except that what it gave us was the capability to, here we just built a number of different gene clusters where we kept the overall organization the same, but at every gene we went from a strong to a low ribosome binding site. And the idea with the algorithms was then to select which combination of ribosome binding sites was necessary in order to optimize function, and you can be very algorithmic about that. And in this case, we got an improvement in activity. So here it's where you're taking a computational method, utilizing your design, build, test pipeline to then use that method to try to better optimize. In this case, it worked in E. coli to improve activity. We also started getting concerned about the way that a transfer would have to happen in reality. So just using operons at all is very constraining. What you would ultimately like to do is move one gene at a time into the new organism and then evaluate where you stand with that. So we said, well, in this system, I mean, there's a lot of talk as to why operons matter. Do they really matter, or can you create a completely, perfectly okay gene cluster with no operons whatsoever? And so in this case, we tested that by creating large libraries where every gene is within its own cistron, so it has its own promoter and terminator. And then to make sure that they really are cistrons, we would flip the orientations back and forth as you go across. And we found some, you lose a lot of activity, but we found some that were perfectly functional, terrible, but perfectly functional. But again, could that then be what we build on? Because to me, as a designer, cistrons make more sense than operons. And they allow you to control the expression level more precisely. So if I could have a fully cistronic system, that would be preferential to me, unless you really need an operon. We went in, we built other ones to see how many cistrons were important, and so on. We've gone in now in a lot of different ways. So we said, well, you know, just having the activity is not enough. So we've gone and used the pipeline to put peptide tags that are cleavable off of every gene, so you can then use mass spec to get all the protein levels. You can put barcodes into the system so that you get all the transcript levels very easily, and so on and so forth. So the whole idea is just progressively going from this messy wild-type system to one that's just as good, but where you've iteratively gone through the process of reorganizing, re-engineering it so that it's measurable, so it's engineerable, so that it can be understood and ultimately transferred. So we were building all of these libraries, and this is getting into the conclusion, and we started to decide that we needed help as a lab. So everything I showed you here is students getting thumb sore putting all these pieces of DNA together. And one of the great things about being at MIT is that we have the sister institute to JGI across the street, the Broad facility, and we decided that this would be an excellent partnership for coupling with them in order to start to apply this at a scale that was just impossible in an academic lab. And you don't need me to tell you about how much manufacturing processes have really accelerated sequencing to a point that was unimaginable 15 or even 5 years ago, I think. And now the Broad Institute is applying these principles to sequence 4.3 trillion base pairs a day. And this was really done by taking all of the molecular biology that was being done in individual labs and converting it into a manufacturing pipeline and going through that in a very rigorous way. And what we're doing then is taking the synthesis and reapplying it to the same system. So that's something that has been up and running for about a month. We have 2 people working there and getting our systems to work in their processes. But I think that once you have this capability where you've got the sequencing, you've got the synthesis, and on top of that you have this element of design, that's going to be a really powerful trio in making use of all of the sequencing information that we've invested so much in. So with that, I'll conclude. I think a big part of understanding genetics is rebuilding the genetics from the ground up. And if you're in genetic engineering at all, if you got nothing from this, think very carefully. You know, when you drop a gene into an expression vector, what exactly you're doing as part of that. And then we have a series of processes here that I think are both helping us optimize the system, understand it better, and ultimately it's going to be important for understanding the underlying biology and ultimately transferring the function. And then I'll just end with, we're not all business. The lab for our Christmas party went around and everybody had to pick a random number between 1 and 64,000, which, as it turns out, you can convert into hexadecimal code, which for nerdy reasons I won't go into, is a minimal sequence to fully specify one of these constructs. So they were, by picking a number, picking a construct, and then they were all assayed, and the winner got a bottle of wine, which is why she's so happy. So with that, I'll conclude. The people that really made this part of the project possible are Michael Smansky in my lab, who came out of Ben Shen's lab, dealing with 100 KB clusters. So for him, it's a downshift. And Dehua Zhao, who came from UCB and was an expert in nitrogen fixation, as well as Karsten Tem. And finally, I'll plug a new journal in synthetic biology that's up and running. And if you haven't seen it, it's worth eyeballing and seeing what's getting published there. So thank you, and I'll take any questions.
Great presentation. My question is that when you go to put it into plants, unlike putting it into E. coli, you need another part, and it's not a part, and that is I can grow anaerobically. How do you plan to, right? You need to protect the nitrogenase from oxygen.
Right. So
how do you deal with that? And given that, have you considered using the superoxide dismutase type of nitrogenase? I'm not as familiar with it, but it requires actually oxygen.
Yeah, absolutely.
I understand. Okay, so thank you.
Oh, did I answer it? No. Oh, okay. Great. So absolutely. So if you're considering moving it into a chloroplast or into a plant, you're going to need the oxygen tolerance. And there are a number of clusters out there from organisms that are able to fix nitrogen in high oxygen environments. They're more complex. The organisms are less well understood. So we started with Klebsiella. But that is the way that you would approach that. What I will say about the Klebsiella one is that it does solve the regulatory part of the oxygen sensitivity in the sense that we can now express whatever we want in high oxygen. It kills the enzyme still because that's at a higher level. But in the native system, everything is extremely tightly off when you're in oxygen. So that aspect's been broken. The other is the ammonia regulation. That aspect is also completely broken. So from a regulatory perspective, what we're doing is we're fixing the genetics so that it can be moved. But any issue with, say, an enzyme or a function like that needs to be solved at a different level. And I think the approach for us is not just looking at individual enzymes that could increase oxygen tolerance, but entire clusters that have that function and they exist. They're just more complex.
Hi. I'm wondering if there's a role for direct selection in any of this, like if you can tie the activity that you're looking for to survival of strains and then use the combinations you've designed to seed that.
Absolutely. So, and in fact, there's a very good selection in this particular case, and we've run some experiments with that. The challenge for us with the selection approach is that you find very specific and often deceptively complex solutions to the problem that you're presenting to the organism. For example, on simpler systems, if we make a ribosome binding site library that's ginormous, and we create a selection so that we can screen that enormousness, you get like a poly A in the ribosome binding site because it's creating some sort of secondary structure in that particular context that allows that level to be achieved. So it's not really by design anymore. It's much more of a specific thing. So we've done some experiments because we were interested in, if you start with the broken refactor version, you run selection, where do the mutations pop up and so on, but we found that was just kind of indecipherable, so we went a different direction to try to increase our knowledge in the system. Yeah.
In many of your synthetic operons, it seems like you have multiple promoters spaced throughout, but in your system, you have your induced versus uninduced strains. So are these orthogonally controlled promoters? And kind of going along with that, do you think it's better to have bigger promoter libraries or ribosome binding site libraries? Because it seems like there's multiple ways to control your overall protein levels.
Right. So for this system, what we have is a single polymerase, and it's a phage polymerase, and all of the promoters are that phage polymerase promoters, and we use promoters of different strengths. In the type 3 secretion system, which you can imagine the same story to the end, for their timing matters. And so we have 2 phage polymerases so that we can attach circuitry to affect the timing of when different operons are turned on synthetically. In terms of what's more important, promoters or RBSs, I don't have a good answer for that, except that one of the reasons we want to destroy operons is that if you have a multi-gene operon, it's largely dictated by the strength of the promoter, of course, but then the first ribosome binding site matters the most. Everything downstream goes from having no effect to like minor tuning. So if you really want to get the levels of all the genes separately, you've got to break it up to have that level of control. And that's our biggest problem now, that we present an internal ribosome binding site as having this strength, but it doesn't at all in that context, and we've got to change that.
I have a bit of a follow-up on that.
Yeah.
Have you actually done transcriptional analysis to see if what's going on is what you think is actually going on in these constructs?
We're setting that up now with the Broad Institute because they've got an RNA-seq pipeline that allows us to do that. I'm anticipating it's going to be a mess.
Yeah,
Yeah.
I would agree with you.
But I'm fascinated by it. And actually, the reason it's a mess is not just that promoters are behaving badly in the wrong spots, but that it's a 94-part system for the full cluster. And these permutations are taking those parts and putting them together in different ways. And so what you have is 93 plus 2 interfaces that are junctions between parts. And so if I have a part with a DNA sequence here, and I got a part with DNA sequence here, and I put them together for the first time, there's a new DNA sequence that crosses that boundary. And trying to make a DNA sequence that does nothing is sort of one of our dreams. And it becomes really challenging because of the information content there to have part junctions that don't do anything. Promoters pop up all the time, terminators pop up all the time, and how you computationally identify them, insulate them, and create parts that don't do that is one of the real leading edge things of what we're doing in SynBio.
They're probably also popping up in those randomized coding sequences, right? You're going to have...
Yeah, most certainly. Yeah, we scan it so we do run promoter calculators and terminator calculators and RBS calculators to find where they might be and get rid of them. But clearly there's stuff that's going to pop through, like the transposon insertion sites that showed up in the first gene. Yeah.
You're ultimately going to need a lot of ATP to run the nitrogen fixation process. Are you worried that you're going to mess up the physiology of the host organism?
Yes. Now you're thinking two steps ahead, but this was a topic that received a lot of discussion at a nitrogen fixation meeting that I was just at, because without all of this, if you just give a plant the ability to fix nitrogen, there's a yield trade-off just because the energy is not going to, and so there's a whole economics around that. And we don't add or subtract anything to that. I will say that if you take it far into the future and think about what you could start doing, I mean, you can imagine pulling in orthogonal systems that are providing the ATP for the nitrogenase. So now, instead of just transferring the nitrogenase, I'm transferring the photosynthetic apparatus from Rhodobacter to couple to it. Right? I mean, that's kind of crazy future, but the possibilities for where you get that energy, if you're able to do all this, it opens up where that might come from. Or you put it in a rhizobial organism, which I think is more realistic anyway. Yeah.
So you are reusing a lot of parts. So I guess there are some parts will have multiple copies, like the promoters. Do you need to worry about the recombination of those repetitive sequence in your
Yeah,
construct?
absolutely. So, and that's to get back to the beginning of the talk, the project I hated, making 700 terminators, was because in the cistronic one, you've got all these double terminators after every cistron, which comes out to about 200 base pairs. You throw that into a REC-A positive strain, and it's going to chew that up like crazy. And so we had to run that big screen just to identify a core subset of 50 strong terminators that won't recombine specifically for that purpose. You know, promoters are not a problem because they're so small.
Right. Well, let's all thank Chris for a wonderful talk.